Hi, this is podcast number 46, and this begins your last week, Principles of Biology 1, where we're going to talk about emergent properties as the last big idea that we haven't introduced a whole lot up until now, and how that important is to biology. And then we're going to give examples of emergent properties, starting with the molecule hemoglobin, which is the protein that carries oxygen in your blood. So if we look at a video provided in your book, as an example of emergent property, if you add Diet Coke and Mentos together, you get this very strong chemical reaction where you have tremendous production of gas. They're just dropping the Mentos into the Diet Coke. So the emergent property is not something you would predict from studying the Mentos, not something you would predict from studying the Diet Coke. It comes out of an interaction with those two things together. Here's another example, former President George Bush and a baby. If you study his properties and his properties, or her properties, I don't know, you wouldn't necessarily predict their interaction, and their interaction, the silly face interaction. You have fear and anger on the face of the baby and surprise on the face of the president. So what about that in biological systems? Why are emergent properties important? Well, life is an emergent property. If you take the component molecules apart in a cell, you can't show where life is, but if you put them together, you get life out of that. And there are many other examples, including this one, about how your tissues transport oxygen. So in large multicellular organisms, you have to have a way of getting oxygen to the mitochondria and getting the waste products, carbon dioxide, out. And so there is a tissue transport system, your blood, that does that. Not all animals out there do that. For example, bugs just deliver oxygen directly through tubes directly to their cells. And that might be a limitation about why we don't have any elephant-sized bugs. But having a transport tissue that allows oxygen to be delivered to cells far away from where you collected the oxygen, that is one key to getting really big as an organism. If you look at the composition of air, it's mostly nitrogen and about 21% oxygen. How do you get the oxygen to the tissues? Well, you could just have a water-based tissue and dissolve the oxygen in water. And this is a graph of how much oxygen you can concentrate in a given amount of water if you want a particular amount of saturation. So if you want it to be 100% saturated, this one here, you can concentrate about 20 kilopascals. It's a unit of pressure into that solution. And a 50% saturated solution, you can put in about, say, 11 kilopascals will give you 50% oxygen saturated solution. That's in a pure water solution. But if we look at what hemoglobin looks like, it's a much different pattern in that if we look before, we said 11 kilopascals will get you about 50% saturated. In this situation, you are over 90% saturated at 11 kilopascals. And it is not a linear curve. It's not related to just how much pressure you apply to how much water. There are different parts of the curve if you're looking at hemoglobin in water. There just to also let you know, not all organisms have hemoglobin. Not even all organisms you're familiar with have hemoglobin. This is an ice fish. It has no hemoglobin in its blood. Here's a vial of its blood, and here's a vial of another fish with red blood. You can see the red color here and the clear color here. There are very few cells in its blood, and there is no hemoglobin in its blood. It's actually clear. This is a juvenile that shows how it's much more clear. But the adults are big, say 20 inches long. They are more milky and not quite as see-through. You do see here the heart. The heart is enormous in these fish for a fish heart. So if this was let's say this is about an 8-inch fish, and it has a heart about the size of the end of my thumb. If this was an 8-inch bass, for example, the heart would be about a quarter of the size of the tip of my pinky. So their hearts are really big, and they just pump a solution of mostly watery blood around, and that blood carries oxygen just as much as the water could carry and no more. So it doesn't have hemoglobin in it to deliver that. How do they get away with that? Well, they live in really cold water. They live in Antarctica, and you can soak more oxygen in cold water than warm water, but not nearly as much as hemoglobin will get you. Okay, so the emergent property we see here is the shape of oxygen binding to this molecule, so that it loads up oxygen in solution where hemoglobin is in a way that you would not predict by simply diffusing oxygen in water. How this is adaptive is that over a very short range of pressures of oxygen, okay, a range of pressures of oxygen that are applicable to living at 1 atmosphere of pressure on Earth, you can get your blood oxygen saturated really high with a fairly small range of oxygen concentrations. And then it's pretty saturated. Normal blood saturation is between 95 and 100%. There's been some talk during the pandemic of people getting a pulse oximeter, which is a little device that you put on your finger, and it takes the color of your hemoglobin as an indicator of how saturated your blood is. They're not perfect, but one thing that people have noticed, some doctors have noticed, is that people's blood oxygen saturation can fall if they're infected with COVID-19 into the 80s, and that's actually really dangerous. Normally, if you are not infected with COVID-19 and your blood oxygen saturation fell that low, you would pass out. And so someone would know something is wrong. But for some people, it falls that low even though they're infected with the virus and they may not even know that's the case. It still has bad effects on the body. And so some people are buying these little devices to check themselves and see if they have low blood oxygen saturation. The other thing about this curve that pertains to our current pandemic is you've heard about people having to be on ventilators. So if their blood oxygen saturation is lower than it should be right around here, 95 to 100%. If it falls low, that can be a problem in terms of just supplying the body with enough oxygen. One way to solve that problem is to provide higher pressure of oxygen. More oxygen supplied to the lungs at higher pressure than you would typically get from just breathing room air. And so people are supplemented with 100% oxygen and sometimes supplemented with oxygen at high pressure to force that into solution into a person's blood if they're infected with COVID-19. And that's the thing about being on a ventilator you've heard about. Okay. So, why is this S-shape? So this is called sigmoidal or S-shaped binding curve. Why is that adaptive? Why was it selected for in the evolution of hemoglobins? Well, if you had a hemoglobin that just had really high affinity, just any little bit of oxygen, it goes all the way up to 100% saturated. Well, that would get oxygen into the blood, but it'd be very hard to get it off of the hemoglobin and into your cells, because then your cells would have to rip it off of that high affinity binding site, and that would be difficult. What if it was really low affinity? Well, then it'd be easy to get it off the hemoglobin molecule when your cells wanted oxygen, but it wouldn't be so easy to get it on. You need fairly high pressures of oxygen out there in the atmosphere, and that wouldn't load up much oxygen. Your pulse oximeter would still say you are really low. So if you have this sort of shape, which is the shape of hemoglobin, you can load up to very high saturation with relatively low concentration of oxygen, okay? And then you're going to see in a second, there's also a way of dumping it off to the tissues. Okay. So, if we look at these three curves, the one in the middle that is orangish, that's the pH of your blood, around 7.4, if you are a mammal, and all of you are. It's a little different if you're cold-blooded vertebrate. It's a little bit higher than that in the lung and a little bit lower than that in the muscle. Now remember, pH is a log scale, so when I say a little bit, it's a little bit on the pH scale, but it's actually a big difference in terms of the concentration of hydrogen ions. So pH 7.4 in your blood, let's say now you want to deliver that oxygen to your muscle, and your muscle has lower oxygen concentration because your muscle has been using that oxygen as a terminal electron acceptor and combining those electrons that came off of stage 4 to make water, and so that oxygen gets used up. You need to supply more oxygen so you can keep on making ATP and using your muscles. So the oxygen concentration, also, as you are metabolizing in your muscles, you're making CO2. CO2 comes off in stage 2 and in stage 3. CO2 dissolves in the water, and that can lower the pH of your muscles. So the pH of your muscle cells becomes a little bit lower, and that means that hemoglobin, when it goes by your muscle cells, also has a lower pH, and as the hemoglobin gets a lower pH, it releases oxygen, okay? So if you look at a given pH at 7.4, okay, let's say we're at this concentration of oxygen, 30 millimeters of mercury. I switched from kilopascals to millimeters of mercury. Sorry about that. But if you're at 30 millimeters of mercury, your blood would have an oxygen concentration of, say, 60%. But at 7.2, pH 7.2, your muscles would only have a blood oxygen concentration of 50%. And so that 10% difference is released from the blood to the muscles and given to the muscles to use. Okay. Similarly, at the lung has a higher pH than the muscles do, and that allows it to carry even more oxygen than in the blood. At a given concentration, your blood, when it reaches the lungs, is going to be close to 100 millimeters of mercury because it's in very close contact with the atmosphere. Okay, and the atmosphere is close to 100 millimeters of mercury, and that will almost fully saturate your blood with oxygen. When it gets to muscle, it's going to be at lower oxygen concentration because the muscles are using oxygen. And that helps a gradient of oxygen, okay? Oxygen is going to want to go from high to low. So it's going to want to go from what came from the lungs to what is going away in the muscles. And at the same time, the muscles have a lower pH, which lowers the carrying capacity of the blood for oxygen. And that causes oxygen to come off of the hemoglobin into the muscle. Okay? All right. So why is this adaptive? Okay. Because you have this sort of situation, you can, where oxygen is in high concentration, you can have very high saturation and pick it up in your lungs. Where oxygen concentration is lower, like in your muscles, because your muscles are using oxygen, you can dump off that oxygen. Plus you have the pH effect on top of that. If you had this shape of a curve or this shape of a curve here in teal, that situation would not work. But because you have this S sort of shape, that situation does work. Okay, and that's an emergent property. You wouldn't necessarily expect that from just looking at the hemoglobin itself. Although we're getting better at predicting emergent properties, and I'll talk more about that in a second. There's also another pigment in your blood called myoglobin. This one here. Myoglobin is one chain of hemoglobin, and it is the oxygen-carrying pigment that is in your muscle. So your muscles are red, both because they are perfused or they have a circulation of blood going through them, but also because they contain this protein myoglobin. If you've ever eaten a tuna steak or seen a tuna steak or a salmon steak at the grocery store, they are very red because they have high concentrations of myoglobin. And what myoglobin does is it binds with pretty high affinity in the muscle. And they are, they're showing you here this curve for myoglobin, sorry. So here's blood, here's muscle, here's the lung, and here is myoglobin. So myoglobin doesn't let go of its oxygen until the concentration of oxygen gets really low. So it has the steeper curve over here, and your oxygen concentration has to get really low toward this end of the curve in order for myoglobin to dump off its oxygen. So myoglobin is like a buffer. It's like an oxygen reserve. After you've gotten rid of the oxygen you can get off of hemoglobin, if you still need more oxygen, then you get rid of oxygen that is stored on myoglobin. It's an extra. In animals that hold their breath a lot, you see high concentrations of myoglobin. So those tuna that we're talking about, they do really deep dives. They're fish, so they can breathe underwater, but they are very, very active, and they are burning lots and lots of oxygen, and so their muscles have a very high demand for oxygen, and that's why they have lots of myoglobin. If you look at a marine mammal, like a whale that is holding its breath and diving, or a elephant seal or something like that is holding its breath and diving, they have very, very high concentrations of myoglobin and very, very red muscles. Same thing for penguins, for example. Okay. This is very little letters, hard for you to look at, but it is an amino acid alignment of the alpha hemoglobins and the beta hemoglobins from lots of different species. Humans, baboons, lemurs, cats, pigs, mouse, shrew, bats, elephants, armadillos, platypus, possums, and chickens. And in an amino acid alignment, there's a computer program that tries to take the one-letter code for a protein, starting with methionine over here, and then line them up with another primary sequence from another species, another ortholog of that same protein from another species, and the computer program tries to maximize the number of amino acids that are the same or similar. And you get this sort of table-looking thing, which is an alignment. And what it allows you to see is what part of the protein has been conserved through the evolution. So at this position right here, this K right here, this amino acid lysine is preserved at amino acid number 41 in all of the vertebrates that are on this alignment. You got to have a lysine at that position. Whereas over here, at position number 79, there's a lot of different amino acids you can have and still be an alpha hemoglobin. In general, you see big blocks of color on this alignment and on this alignment. So in general, these are considered very highly conserved sequences. If you saw big blocks of white, like here, they would not be considered highly conserved sequences. So what's the message there? The message is that don't let evolution mess with your hemoglobins dramatically, otherwise you won't survive. So it's not that these hemoglobins haven't accumulated mutations, it's just that if there was a mutation that didn't allow the hemoglobin to work, that individual didn't survive. And so only the sequences that allow the hemoglobin to work have survived, and they are pretty much the same sequences all the way from chickens to humans. If you look at the sequence information and how that tells you how these proteins are related to each other, for one of the sequences, it put platypus and chickens closer together than, and in the beta hemoglobins, it put possums and chickens closer together. And also the position of the pig. Pig are closer to shrew in the alpha hemoglobins, and pig are closer to cats in the beta hemoglobins. So that's not super unusual. Again, these are how the proteins are related to each other, not necessarily how the animals are related to each other, although the two should be pretty close to each other, the two types of trees. But it's just an example of how you can use phylogenetic trees to see how proteins are related to each other, just like you can use phylogenetic trees to look how species are related to each other. Okay, so I said that hemoglobin binding oxygen in the way that it does is an emergent property of the molecules. If you put the amino acids together in a particular way, you wouldn't necessarily be able to predict that it had these properties. And part of its emergent property is its ability to bind oxygen cooperatively. So that has to do with how the molecule moves and flexes when it binds oxygen. So this is the heme. These gray balls here are the heme in the middle of the protein that contain the iron molecule that gives your blood its color. And this iron molecule is going to move. You're going to see whether it does or does not have oxygen bound to it. So this is no oxygen bound to it. This is with oxygen bound to it. Again, no oxygen with oxygen. You can see that iron molecule moves down in proportion to how you're looking at it. That is connected to the rest of the protein structure. And so when iron binds, it actually changes the shape of the protein. Okay. Here is those heme molecules within the rest of the hemoglobin structure. 1, 2, 3, 1, 2, 3, 4 of those. And the beta sheets are in pink here, and the alpha sheets are in green here, each one with a heme. And as this iron moves, it causes the whole thing to flex. And as one oxygen is bound, it makes it more likely for the second one to be bound, for the third one to be bound, and for the fourth one to be bound. That's called cooperativity, and you see that a lot in protein structures, and it's an emergent property of protein structures in that they cooperate in the sense of one binding one ligand, one thing that it's going to bind, makes it more likely to bind a second one, a third one, and a fourth one. Okay. Here's another view of with or without oxygen and how it changes the shape of the whole protein when oxygen is bound or not bound. So that cooperativity is coming from this shape change, and that emergent property comes from this shape change. So even if you had all the amino acids together, we're not at the point yet where you can predict for a protein that it would necessarily have this cooperativity. That might not be true in the last couple of years. So in the last 10 years, you weren't able to predict the 3D structure of a protein just by looking at its order of amino acids. We've actually gotten lots better at that since then, and there are computer simulations that may be able to predict this sort of thing just from looking at the amino acids. But we still consider it an emergent property. Okay. So, what happens to the oxygen and how? Here's this little red blood cell. Red blood cell looks like a lozenge. Red blood cells, and when they become mature, they actually kick out their nucleus so they can pack in more hemoglobin. And over 99% of the protein in a red blood cell is hemoglobin. So it's essentially a big bag of hemoglobin. At the lungs, there's relatively high pressure of oxygen because the lung cells are in very close contact with the atmosphere from breathing in oxygen. That's where oxygen is going to get picked up, has a very high affinity for oxygen at that point, and most of your oxygen is going to be, or most of your blood is going to be saturated. Most of the hemoglobin molecules will have oxygen bound to them. That's what your blood being saturated means. And it'll be at a higher pH in your lungs. As it travels to the muscles are using up oxygen, so the concentration of oxygen available is much higher in the lungs than it is in the muscles. Muscles are using oxygen, so the concentration of oxygen is lower. In the blood outside the muscles, that blood came from your lungs, and it has a high concentration of blood bound to hemoglobin, and there's low concentration of oxygen in your muscles, and so the oxygen is going to want to go high to low because there's a gradient there, and because there's a fairly steep gradient, that's the way oxygen is going to want to go. At the same time, your muscles are metabolizing and producing CO2. CO2 dissolves in the water of your muscle cells and in your blood, and it lowers the pH of your blood. And blood at a lower pH means that hemoglobin can carry less oxygen even if oxygen is around. So its ability to soak up oxygen goes down as the pH goes down, and so that releases oxygen off of the hemoglobin, which is used by the muscles. Okay, these are emergent properties of this hemoglobin molecule that it has cooperative binding. Once one heme binds an oxygen, it's more likely for a second one and a third one and a fourth one to bind because of this oxygen binding causes a shape change in the protein. And then also that the hemoglobin molecule is pH sensitive. And as the pH lowers in the muscles, more oxygen is released. Then it goes past the muscles back to the lungs, pH increases, and it can pick up oxygen, and around we go.